Plant density. Counting methods. Density is defined as the number of individuals per unit area. Density reflects the closeness of individuals to one another, and counting methods are among the most common approaches used to measure density. Counting methods are plot-based, meaning that they occur in areas that are delineated by borders, so they have edges. Quadrats, or frames, such as these that you see in the photo here, have hard edges, and they're constructed of steel, aluminum, PVC pipe, sometimes wood, and occasionally flexible plastic. These quadrats can assume different shapes, so we can use square, rectangular, or circular quadrats. And we usually use frames when the size of the quadrat that we need to use is relatively small, usually no larger than 1 meter squared or possibly 1.5 meters squared, because they need to be carried around out in the field. When we need larger plots, we often establish those using transect tapes and then staking the corners of the plots, making sure that the plot is the correct dimensions, and then we sample within these areas. As you can see, plot sizes can be quite variable, and sometimes we choose to use very long plots with narrow widths, and in this case we have a special term for those plots. We call them belt transects. Now let's look at the basic calculation of density using a counting method. The units for density are the number of plants per unit area, and we're going to look at an example where we want to estimate the density of the pink daisy in this schematic diagram here. We're going to use 3 1/4 meter squared quadrats that are randomly located, and you can see by their placement that 2 of the quadrats have 1 daisy in them, and 1 of the quadrats, the center one, does not have any daisies. So the counts per quadrat are 1, 0, and 1. Now in order to calculate, if we wanted to determine the number of plants per meter squared, we would first divide the total number of plants by the total number of quadrats, and so we would get 0.66 plants per quadrat. But remember, the quadrats are 1/4 meter squared, so we have to adjust for that and multiply by the size of the quadrat. So 1 quadrat was 1/4 meter squared, and so this results in an estimate of approximately 2.7 plants per meter squared. Now it's important to point out that this example is just for the purposes of showing how density is calculated, and we would almost never use a sample size of only 3 quadrats to estimate density, and we'll talk about that a little bit more later. When we are using this method, it's always important to clearly state your ground rules, and the first ground rule that needs to be determined is what is the counting unit. So we need to identify the counting unit. Is it an individual, such as we see this individual bunch grass here? Or if we do want to use individual plants, sometimes we can encounter situations where it's very difficult to determine what is an individual, especially when plants are clonal, if they reproduce vegetatively above or below ground, or sometimes for plants that are very closely spaced. Now sometimes it may not be necessary to identify and delineate individual plants, depending on what our management objectives are. And so it may be sufficient simply to count plant parts that we're most interested in. Oftentimes we will do this looking at stems, tillers, inflorescences, or whatever the plant part of interest is that is going to best provide information for the management objective. Another ground rule is that it's important to know when to count a plant as inside the quadrat, because remember, the density is the number of plants per unit area, and the plants need to fall inside the density. The general rule is that we use rooted density. A plant needs to be rooted inside the quadrat in order to be counted as in. And it could have canopy that extends outside of the quadrat, but as long as it's rooted inside the quadrat, it would count. In the situation where we have plants that are not rooted inside the quadrat, even if they have canopy that extends over the edges of the quadrat, they would not be counted as in the quadrat. Okay, with density and counting methods, we also have to deal with boundary decisions. Because we're working with quadrats and plots that have edges, we need to make decisions when plants are touching or intersect the edge of the quadrat. And some of the dilemma is described here. What if the stem touches the edge, such as here, or there's a stem touching the edge of the quadrat here? What if less than 50% of the stem is inside the quadrat? Or what if more than 50% of the stem is inside the quadrat? And then what would you do if it's exactly half in and half out? Well, we solve this dilemma by establishing clear decision rules. And there's a couple of ways of going about this. One rule is the 2 sides in, 2 sides out rule. And essentially what you do is you take your quadrat, so we see a quadrat frame here, and you arbitrarily decide that perhaps the top and the left would be considered in. So anytime you have to make a decision about a plant, if it touches the left side of the quadrat or the top, you would count that plant as in. And in order to balance this out, you would then say that any plant that touched the bottom edge or the right edge would be counted as out. Anytime you have to make a decision, so you don't have to concern yourself with how much of the stem or is in or out. Another way to do it is to simply alternate in versus out. So the first time you encounter a plant that you have to make a decision about, you would count it as in. And the next time you encounter another plant that you have to make a decision about, you would then say that it is out, and you would alternate in versus out. And this balances out the difficulty of having to spend time determining whether a plant is in or out, and also reduces the chance for any kind of observer bias. Now, there's another approach that some people will take, and that is to examine carefully each case and decide on a case-by-case basis whether a plant would be in or out. But in general, this approach is not favored for a couple of reasons. One is it is very time-consuming, and also there is the potential for the observers to introduce non-sampling error or bias. The next set of decisions we need to make are in regard to selecting the appropriate sampling unit. Remember, our sampling objectives are that we want to have a precise and accurate estimate, and we want to have the greatest efficiency possible for a given sampling effort. And so with counting methods, it's important to determine what is the appropriate size and shape of our quadrats or plots. These decisions are based on the average plant size that we're focusing on, as well as plant dispersion. So we're interested in whether the plants are homogeneously distributed or relatively evenly distributed throughout the landscape. Sometimes plants can be heterogeneously distributed, where they are patchy or clumped. And sometimes we can encounter a spatial gradient in the distribution of plants. All of these factors influence our decisions on size and shape. So let's take a little closer look at that, first focusing on decisions about size. Now, if we start out with relatively small quadrats relative to the average size of plants or the amount of space between plants, we often can run into situations where we will get a lot of empty quadrats. But we can see that if we keep the location of the quadrat the same, but simply increase the size of the quadrat, that we will start to have quadrats that include more plants and fewer that are empty. The general guidelines are that we want to have our quadrats size selected so that it's larger than the size of the average plant. We also want the quadrat size to be larger than the average space between plants. Now you can see here that we should start to consider that this is different for different types of plants. The average size of the green plants and the space between them is very different than the average size of these purple plants and the space between them. And so oftentimes it's helpful to have different quadrat sizes for different species of plants. Let's next focus on quadrat shape. If we start off with square quadrats, we often can find in patchy vegetation that we may have more empty quadrats or quadrats that don't really encompass the variability of the site. But as we elongate the quadrats, we will be able to incorporate more plants and cut across the spatial variability of the vegetation. So in patchy vegetation, we often will move to more rectangular, elongated quadrats because they cut across these clumps or patches of plants. And this tends to increase precision in sparse vegetation. Now let's consider the effect of size and shape on precision. First, we'll look at size. In this example, we see a population, two schematics of the same population. Plants are represented by these little X's, and we can see that they're how they're distributed in the landscape. And the true population here is 0.8 plants per meter squared. We're using square quadrats, but of different sizes. And so here we're going to compare the effects of going from a smaller quadrat to a larger quadrat on the precision of our estimate. We're using a sample size of 5 for the ease of the demonstration. So if we assume that the small quadrat is 1 meter squared and the large quadrat is slightly more than twice as large, 2.25 meters squared, we can see with the small quadrat that we have several quadrats that are completely empty. And this is not an ideal situation. It tends to decrease the precision, so we have a relatively high or large standard deviation relative to the size of the mean. The standard deviation in this case is actually larger than the mean. On the other hand, when we increase the size, we start to have more plants incorporated in the quadrats, and we can see that there's fewer zeros and that the size of the standard deviation decreases. So this ultimately increases the precision of the estimate. Now let's look at the effect of shape. Here we have the same population, and we are using a sample size of 5 to estimate density. We're comparing the square 1 meter squared quadrat to a rectangular quadrat that is the same size. It's simply different in shape. And here's what we see. Again, we have the same information from before, several empty plots with the square quadrat. But with the rectangle, we're keeping the quadrat based at the same point, we tend to pick up more of the spatial variability. We have fewer quadrats that are empty, and this effectively reduces the standard deviation and increases the precision. It's important to note again that a sample size of 5 is generally too small to estimate density in a real world situation, but this is for the purposes of demonstration. Now let's talk about nested quadrats. Remember, I briefly mentioned before that when you are focusing on different size plants, such as these that we see here, or plants that have different distributions, we often find that the ideal size of the quadrat for different species is different. And we can figure this out by doing pilot sampling and seeing what size provides the most precise estimate for different species through our pilot sampling. And as we can see in this example here, we have determined that the grass plant, because it's far smaller, it's also evenly distributed, it really only needs a smaller quadrat. So we've got that in this yellow quadrat, and every time we would set down a nested quadrat, we would measure inside the small quadrat, inside the medium quadrat, and inside the largest quadrat. The way this is done is you would place the quadrat down. Inside the yellow quadrat, you would only record the number of grasses. Inside the blue quadrat, you would ignore the grasses and the thistle, but you would only record the daisies. And then inside the largest quadrat, which is the orange quadrat, you would use those to count the thistles. It's important to note that the blue quadrat includes this portion of the yellow quadrat. So the mid-sized quadrat includes this portion of the smaller quadrat, and the largest quadrat includes the entire area, so it includes the plants that are in this portion as well. Remember that the plants are measured simultaneously with a placement of each nested quadrat. So here we see a couple of schematics. In general, what we do is we use different sizes and sometimes different shapes depending on the situation and what our management objectives are. So when we're interested in different kinds of species or functional groups, or sometimes we will also use this if we're interested in age classes of the same species. So for example, in this large plot here, if we assume that we're measuring a woody plant and we want to know information about the density of seedlings as well as saplings or juvenile plants and mature plants, it would be very time consuming to count seedlings in something particularly large. So you would have the smallest quadrat, these little squares in the corner, would be used to count seedlings. Your juvenile or saplings would be measured in a belt transect, and then the larger plot would be used to count the mature plants. So that's one application of using nested quadrats. This is a very efficient approach to use when you're interested in different species, especially with different sizes and distributions. Now there are fundamentally some guidelines that we use when we're trying to determine the number of quadrats as well as the size and the shape. First, it's important that we conduct pilot sampling. Based on our pilot sampling, we would then increase the plot size in general when more than 20% of the plots are empty, completely empty. And we would also increase the plot size if the number of empty plots is greater than the number of plots with one or more plants. There are some guidelines that in general we want to have multiple plants in in each plot or in the majority of plots, but that is very dependent on the type of vegetation that we are measuring in, the kinds of plants, and their distribution. Uh, so it's important to always consider your site-specific characteristics as well as management objectives when determining the appropriate plot size and plot number. In sparse vegetation, we tend to use longer plots that have been elongated because they will cut across the variation or the variability in the vegetation. And we use nested quadrats if species of interest require different plot sizes. It's always important to clearly state our ground rules before initiating sampling efforts.